Good morning, my dear student friends. Today we will continue the chapter Sexual Reproduction in Flowering Plant. Last class we have started. Last class we have started the Biology Brahmastra series, part 1. Today we are going to discuss the second part of Biology Brahmastra. तो आज थोड़ा ज़्यादा एनर्जी है, चलो स्टार्ट करेंगे क्या? तो लास्ट क्लास I have told you about different types of flower. That is your bisexual flower or hermaphrodite flower, another is unisexual flower or imperfect flower, another is a neuter flower. So today we are going to discuss about type of flower on the basis of a number of carpel. Number of carpel के ऊपर बेस करके. Carpel क्या है? Last class I had told you what is carpel. If you look at the flower, the stalk of the flower is called pedicel. This is the pedicel, and the upper end of pedicel is known as thalamus. Over the thalamus, first is sepal, second part is petal, third part is stamen, and last part is carpel or pistil. This carpel or pistil, and the carpel or pistil is the female reproductive part which forms the gynoecium. This is called carpel. Carpel or pistil, carpel or pistil, female part. Clear? तो on the basis of this number of carpel or pistil, pistil को देखकर carpel or pistil कितना है, how many number of carpel or pistil are present in a flower, on the basis of that flowers are two types. First, it's called a monocarpellary flower, monocarpellary flower. Another second is called as a multicarpellary flower, multicarpellary. What is the difference between monocarpellary and multicarpellary? Listen carefully. Mono means one. If a flower contains only one carpel, that is called as a monocarpellary flower. And if a flower contains more than one carpel, that is called as a multicarpellary flower. So in this diagram, what you find in this flower, how many carpel is present? Only one carpel. So this is one example of a monocarpellary flower. And if you get more than one carpel, that is called as a multicarpellary flower. This is the basic difference between monocarpellary and a multicarpellary. But if you come to this multicarpellary flower, this second one, monocarpellary is clear to all. If you come to this multicarpellary flower are again categorized into two groups. Multicarpellary flower are again divided into two types. And what is the difference? Why they are divided into two types multicarpellary? When many carpels are present, two conditions are exists. First condition, sometimes in case of multicarpellary flower, in some cases you find many carpels are fused with each other or united with each other like this. Sometimes you find all the carpels are fused with each other. This is the carpel number 1, 2, 3. All the three carpels are fused with each other. But sometimes it is found the carpels are free from each other. Carpels are not united. Carpels are free from each other. Now you can differentiate this condition. In this case 3 carpel, in this case also 3 carpel. 3 carpel means not one. When one carpel is present, monocarpellary. If there is a single carpel, it is called monocarpellary. But here more than one carpel, that is called multicarpellary. In multicarpellary, again two conditions arises. Sometimes all the carpels are fused. Sometimes all the carpels which are present in multicarpellary condition, all the carpels are free. And when all the carpels are fused, that is called as a syncarpous condition. Syncarpous, very important question. Comes in NEET as well as in your board. What is a syncarpous? In multicarpellary flower, when all the carpels are fused, that is called syncarpous. And in multicarpellary flower, when all the carpels are free, that is called apocarpous. These two important terms you have to remember. Syncarpous condition, all the carpels are fused. Apocarpous condition, all the carpels are free. and in all cases examples are there. Examples are not important for your board examination. Examples are important for your entrance. What are the examples? I am writing the example of syncarpus condition. Syncarpus example is papaver, poppy plant, papaver. Another is mustard, another is tomato. And how to remember these examples I will tell you, but in offline class, not in this class. Okay, so here you have to only listen. The formula I will not tell you. No formula will be given in the online class. So syncarpus, papaver, mustard, tomato. But if you come to apocarpus, example of apocarpus is lotus, rose and michelia. These are example of apocarpus flower. So syncarpus and apocarpus two condition complete. But very important is their definition. When the carpels are fused, that is called syncarpus. When the carpels are free, that is called apocarpus. And a note, PDF note of this chapter, not this chapter, entire your 12th, I have given in PDF form. Okay, so you have to read that and go through this lecture. Okay, so now syncarpus and multicarpus complete. Monocarpellary, what is the difference between monocarpellary and multicarpellary? That part we have completed. So types of flower depending on number of carpel. This part is clear. Now we'll move to the next part. Next important part, just like we have discussed the fused condition of carpel and a free condition of carpel, syncarpus and apocarpus condition in flower. Another classification, not classification, is condition. Just like syncarpus and apocarpus, we have seen just recently. The last class I had told you, if you look at a flower, so what are the parts present in a flower? This is the pedicel, stalk of the flower, and just stalk of the flower, the upper end of the stalk is called as thalamus, and over the thalamus first part is the sepal, and a sepals form a hole. I had told you these sepals forming one hole, that a hole is called as a calyx. Similarly another hole is there, petal. These petals forming another hole, that is called as corona, not corona, it is corolla. Okay, corolla, calyx, it is corolla. So next, after that there is stamen, and last is carpel. Okay, so these are the important parts. So about carpel I had told you just recently. So but now come to these parts. First part is called sepal. Sepals together form calyx. Similarly second part is called petal. Petal forms corolla. So now come to the condition. If you look at this sepal, first part, sepal, the calyx is made of sepal. Each unit of calyx is known as sepal. But inside the calyx you find two conditions. Just like in carpel I have told you, in multicarpellary there is two conditions, syncarpus or apocarpus. Similarly in case of calyx, if you look at the sepal, sometimes all the sepals are fused with each other like this. Here I have shown three sepals. All the sepals are fused. Sometimes the three sepals remain free. They are not fused with each other. First, second, third. So what is the difference between these two conditions? Here sepals are fused with each other. Here sepals are free. And when the sepals are fused, united, it is called as a gamosepalous condition. Gamosepalous condition. And when sepals are free, that is called as a polycephalous condition. Polycephalous. These terms you have to remember. In case of carpel, we have used syncarpus and apocarpus. But in this case, you have to use, in case of sepal, you have to use gamosepalous and polycephalous. When sepals are fused to gamosepalous, when sepals are free, that is called polycephalous. Similarly, I can take example of corolla. Calyx is made of sepal, but corolla is made of petal. So again there is two conditions. In case of petal, you find sometimes petals are united or fused with each other. When all the petals are united, that is called as a gamopetalous. 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 Similarly, when all the petals are free, that is called as a polypetalous. Polypetalous. So gamo and poly is used for sepal and petal. When they are united, gamocephalous or gamopetalous respectively. When they are free, polycephalous or polypetalous respectively. Clear? So about condition of sepal and petal we have completed. Now come to next part. In some flower, in this flower, you can clearly observe there is sepal and another part is petal or calyx and corolla. But some flowers are there where the calyx and corolla are not distinct. I had told you in the last class, the calyx is generally green in color. What is the color of the calyx? The calyx is generally green in color. This part is green in color. And what is this part? This part is brightly colored, maybe red, yellow, orange according to the type of the flower. But this distinction in color is found in maximum flower. But some flowers are there where the distinction is not present. For example, in case of lily and onion, and lily, some plants are there where the flowers are not having distinct sepal or petal. I am showing one diagram. This is one flower. This is the pedicel, this is the thalamus, and this is one part. I am showing this is one part of the flower, and this is stamen, and this is carpel. If you look carefully, if you look at this flower carefully, if you compare with this flower, in this flower, calyx and corolla are completely distinct, completely recognizable. Yes or no? The calyx and corolla are completely distinct. You can distinguish according to color. This is green in color, this is maybe red in color or yellow in color according to type of the flower. But here there is no calyx and corolla. Calyx, corolla are combined. In this case, you can find stamen and carpel are different, but calyx, corolla are not clearly distinct. When calyx and corolla are not distinct, that whole lily is called as a perianth. You have to remember, perianth. That whole means complete. The complete whole is called perianth. Here sepal petal is each part, but that structure, that whole is called as calyx. This whole is called calyx, this whole is called as corolla. Similarly, here calyx, corolla is not present. Calyx and corolla. So what is perianth? Sometimes question comes. Perianth is nothing when calyx and corolla are not distinct. When calyx and corolla are distinct, they are used as calyx and corolla separately. But when calyx and corolla are not distinct, better I can show in this side. When calyx, because I have written here calyx and corolla this side. So when calyx and corolla are not distinct, not separate, that is called as perianth. So this structure is called as a perianth. Just like calyx is made of each unit. ये कैलिक्स का एक हिस्सा, एक पार्ट को क्या बोलते हैं? सेपल. कोरोला का each पार्ट को क्या बोलते हैं? पेटल. Similarly, if you come to this perianth, in some flower, when perianth is present, it represents both calyx and corolla. Their unit is called as a tepal. Each part is called as a tepal. Each part is known as tepal. तो यहाँ पर हम ऐसे बोल सकते हैं, सेपाल को मिला के बनता है कैलिक्स, पेटाल को मिलके बनता है कोरोला, टेपाल को मिला के बनता है पेरियांथ. तो पेरियांथ इज द होल, बट ईच पार्ट इज कॉल्ड एज ए टेपाल. ईच यूनिट ऑफ पेरियांथ इज नोन एज टेपाल. तो व्हेन कैलिक्स एंड कोरोला आर एब्सेंट, at that place you find perianth. So this completes your different types of flower and their parts. Okay, now you can take a screenshot of this, or you can write in short. This part is clear perhaps to all of you. So now different types of flower we have completed. Now we'll move to the next part. So these are some important 1 mark questions which may be asked in your exam. Very important for examination point of view. So next we'll move towards another important part, that is stamen. So today we'll discuss with male reproductive organ. Last class I had told you Androecium is made of stamen. So today we will start with the stamen. Stamen is called a male sex organ or male reproductive organ of a flower, and carpels are known as a female reproductive organ of the flower. So stamen, this male reproductive organ. If you look at the stamen, what a stamen contains? Each stamen consists of, if you look at the stamen, each stamen consists of two important parts. Each stamen consists of two important parts. This upper part is called as anther. This part is called as anther. This part is called as anther. And this long and slender part, this part is called as a filament. So two portions are there, anther and filament. This part is called as filament. Anther and filament. Two parts. Anther is a lobed structure. It is a lobed, maybe spherical or oval, egg-like. So this anther is the terminal part. Terminal means end. And this long and slender stalk-like structure is called as filament. ये दोनों को मिला के बनता है stamen. Stamen is made of these two parts, anther and a filament. And the point at which anther and a filament is joined, there is a special tissue. That tissue which connect anther and a filament, that is known as a connective. That is known as connective. So connective is a tissue which connect anther and a filament. So anther is the terminal part. And if you look at the anther, each anther contains two sides. So it is a bilobed structure, bilobed. One is left side, another is right side. So two lobes are there. Just like in heart, there is four sides, four chambers. Similarly, here there is two sides. These sides are called as a lobe. So if you cut it transversely, अगर हम एक आंथर को ट्रांसवर्सली ऐसे कट कर दे, ऐसे कट कर लेंगे ट्रांसवर्सली, तो इसका ऊपर पोर्शन को निकालने के बाद क्या मिलेगा? हमको दो साइड मिलेगा. तो अभी उसको हम डायग्राम में शो करेंगे. Each anther, if you look at the anther, each anther consists of two sides. So listen carefully. Each anther consists of two sides. This is anther and the lower part is filament. This part is filament. Okay. This part, this is the long slender filament, and this part is anther. This part is anther. After cutting, we are discussing the section. This is the section. After transverse section, ये ऊपर वाला पोर्शन हटा दिए. Okay. कट करने के बाद तो क्या मिलेगा? हमको दो साइड मिलेगा. This is one side. This is another side. Upper portion is already removed. Transverse section. So this is the lower half portion, lower half of this anther. Lower half of the anther is shown in this diagram. So in this diagram, how many sides you are looking at? This is one side, this is another side. Each side is called as a lobe. इसको anther lobe बोलते हैं. Anther का lobe. So this is one anther lobe. This is another anther lobe. So how many anther lobe you find? Two anther lobes. These are called as anther lobe. Anther lobe. Anther lobe. So how many lobe you find inside anther? There is two lobes. So that's why anther is a bilobed. Anther को क्या बोलते हैं? Anther is a bilobed structure. We can say it is having two lobes. Bilobed. Bilobed structure. Anther is a bilobed structure. Anther is a bilobed, two lobes. But if you enter inside each lobe, there is some circular structures, two circular structures in each kernel. In this kernel, there is a circular. After cutting, you find a circular structure in this side, another circular structure in this side and in this side. So that's why, if you look carefully, in each anther lobe, there is a two structures, and these structures are called as a microsporangium. Microsporangium, क्या बोलते हैं इसको? Microsporangium. Microsporangium. This structure is called as a microsporangium. 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 So two microsporangium are found in each anther lobe. But in total anther, in a single complete anther, you find 4 microsporangia. So that's why anther is tetrasporangiate in nature. Each anther is tetrasporangiate. Tetrasporangiate. You have to remember what is the meaning of tetra. Tetra means 4 sporangiate, microsporangium. So microsporangium is nothing. Remember this. Microsporangium is nothing. Microsporangium is a circular structure. After transverse section, it is a circular looking structure under microscope found in 2 number in each anther lobe. So that's why at the 4 corner, this corner, this corner, this corner, this corner, at 4 corner there is 4 microsporangium. And these microsporangium, when develops further, they converted into pollen sac, which produces pollen grain. You might have heard in lower classes, 9th or 10th classes, what is pollen grain. During reproduction of plant, some pollen grains are produced. So this microsporangium changes into pollen sac. ये माइक्रोस्पोरेंजियम चेंज होके क्या हो जाते हैं? पोलन सैक. तो माइक्रोस्पोरेंजियम चेंजेस इनटू पोलन सैक. पोलन सैक्स. तो पोलन सैक्स आर नथिंग. पोलन सैक्स आर नथिंग. This same microsporangium. This same microsporangium which are present when they become mature, they become pollen sac. So that's why now question comes: how many pollen sac you find inside each anther? Each anther contain 4 pollen sac. Each anther lobe contain 2, but total anther contain 4 pollen sac or 4 microsporangium. Clear? तो microsporangium क्या होता है? Microsporangium in the beginning, but when they become mature, after maturation they become pollen sacs. So that's why anther is bilobed but tetrasporangiate in nature. Bilobed but tetrasporangiate in nature. This much you have to remember. Then we'll move towards the next part. How this anther is formed. ये जो anther अभी देखे, ये anther कैसे formation हुआ? How the anther develops. How this anther develops. How formation of anther takes place and formation of this microsporangium takes place. This part, up to this part, I think everybody you are clear about these things. Now come to next important part, development of anther. How anther development takes place. Development of anther. Development of anther. ये anther कैसे formation होता है? Development of anther. Development of anther. ये anther कैसे formation हुआ? How anther develops? Initially, when a flower is formed, when the flower is formed, the anther is not there. So what is there? Initially there is only group of cells. Some group of cells, and these cells are highly divided. Initially there was no anther. Anther कभी भी नहीं होता है. Initially क्या होता है? जो anther दिखाया अभी, दो लोब था और माइक्रोस्पोरेंजियम 4 थे, 4 कर्नल में, इसे कुछ स्ट्रक्चर नहीं होता है. Initially क्या होता है? Group of cells, and this group of cells are meristematic cells. Meristematic. What is the meaning of meristematic? In 11th, first year you have read. Meristematic means highly dividing cells. Those who can divide. Meristematic cells. And these all are homogeneous. Homogeneous means same size and of same character. The meristematic cells. And they are covered by one covering known as epidermis. उनको कवर करके क्या होता है? एपिडर्मिस. एक कवर को क्या बोलते हैं? एपिडर्मिस. तो इनिशियली दिस इज द स्टामेन, दिस इज द आंथर, नॉट स्टामेन, दिस इज द आंथर. स्टामेन कंसिस्ट ऑफ 2 पार्ट्स, आंथर एंड फिलामेंट. तो इनिशियली आंथर इज लाइक दिस. आंथर के अंदर कुछ भी नहीं मिलेगा. नो लोब, नो पोलन सैक्स और नो माइक्रोस्पोरेंजियम. इनिशियली आंथर इज ए मेरिस्टेमेटिक ग्रुप ऑफ मेरिस्टेमेटिक सेल्स कवर्ड बाय एपिडर्मिस. लेकिन बाद में क्या होता है? जब ये धीरे धीरे बढ़ता जाएगा, थोड़ा थोड़ा चेंज होता जाएगा इसके अंदर. चेंज क्या होगा धीरे धीरे? तो इसको अभी हम एक ट्रांसवर्स सेक्शन लेके देखेंगे. ट्रांसवर्स सेक्शन, इसका ट्रांसवर्स सेक्शन ले जाएंगे. एक्चुअली दिस इज ए स्फीयर. ओके, दिस इज इन सेक्शन वी आर डिस्कसिंग. दिस इज ए सर्कल फ्रॉम साइड, बट एक्चुअली इट इज ए स्फीयर. You can imagine this group of cells inside a balloon. there is a balloon sphere, okay, sphere or oval structure, inside that many cells are present. तो these cells are present. अगर इसको ट्रांसवर्स सेक्शन हम ले जाएंगे, क्या मिलेगा? ट्रांसवर्स, ऐसे ट्रांसवर्स ले जाओ, ट्रांसवर्स सेक्शन कर लेंगे तो क्या मिलेगा? एक सर्कुलर लाइन मिलेगा, सर्कुलर आउटलाइन. और एपिडर्मिस, एपिडर्मिस तो चारों तरफ है, तो हमको कैसे दिखाई देगा? आफ्टर ट्रांसवर्स सेक्शन ऐसे दिखाई देगा क्या? नीचे तो फिलामेंट है, यहां पर फिलामेंट नहीं दिखा है, ऐसे फिलामेंट होगा नीचे, ओके, तो ऐसे दिखाई देगा. तो अभी मैं अपर से सेक्शन ले गया, इसको साइड से दिखाया था, लेकिन अभी क्या किया? अपर सेक्शन ले गया. तो सेल कैसे दिखाई देंगे? सेल इस साइड में अभी दिखाई नहीं देंगे, सेल यहां पर दिखाई देंगे अपर से. तो दिस इज द होमोजेनस सेल्स कवर्ड बाय एपिडर्मिस. लिसन केयरफुली, वेरी इंपोर्टेंट पार्ट. दिस इज द एपिडर्मिस. एक कवर को क्या बोलते हैं? एपिडर्मिस. दिस कवरिंग इज कॉल्ड एज एपिडर्मिस. Covering is called epidermis, and there is a mass of cells. These cells are highly dividing, called as meristematic cells. Okay, these all are the cells. तो इसको अभी ट्रांसवर्स सेक्शन लेके हम माइक्रोस्कोप में देख रहे हैं. तो अभी क्या होगा धीरे धीरे? तो पहले तो ये ग्रुप था, मास ऑफ सेल्स, और कुछ नहीं था. लेकिन थोड़ा रिकॉल करो जो डायग्राम में बनाया था, यहाँ पर जो आंथर का डायग्राम दिखाया था, दो साइड था, उसमें एक साइड में क्या क्या थे? दो माइक्रोस्पोरेंजियम थे. यहाँ पर कुछ दिखाई दे रहा है? ऐसे कुछ भी नहीं दिखाई दे रहा है, क्योंकि ये वाला इनिशियल स्टेज में है. इनिशियल, तो दीज आर द इनिशियल स्टेज. इनिशियल मीन्स स्टार्टिंग स्टेज. इनिशियल आंथर, दीज आर द इनिशियल आंथर. अभी हम देखेंगे कैसे डेवलपमेंट होगा, डेवलपमेंट ऑफ आंथर, कैसे आंथर मैच्योरेशन स्टेज को पहुंच जाएगा. तो पहले क्या होगा? एक एपिडर्मिस होता होगा इनिशियली. तो धीरे धीरे क्या होगा? दिस सेल्स बिलो प्रेजेंट जस्ट बिलो द एपिडर्मिस. This cell present just below the epidermis. अभी थोड़ा देखो, just below the epidermis का मीनिंग क्या है? This is the epidermis. This is the epidermis. Epidermis से थोड़ा नीचे चले आओ, थोड़ा नीचे, epidermis से थोड़ा नीचे. तो कौन सा सेल मिलेंगे? यहाँ पर इस सेल, this cells. ओके, तो अभी क्या होगा? This cell present just below the epidermis in vertical rows. In vertical rows at the four corner. चार कर्नर को ले जाएंगे, चार कर्नर. यहां पर एक कर्नर चूज कर दो. दिस इज फर्स्ट कर्नर, दिस इज सेकंड कर्नर, दिस इज थर्ड कर्नर, दिस इज फोर्थ कर्नर. मैं चार कर्नर भी चूज कर दिया. लेकिन ये जो अपर वाला सरफेस में दिखाया, एक कौन सा सरफेस? आफ्टर कटिंग ट्रांसवर्सली. लेकिन सेल क्या ओनली यहां पर होते? नीचे नहीं होते, नीचे सेल होते हैं. नीचे कैसे सेल होते हैं? ऐसे रो में. तो देयर इज मास ऑफ सेल्स. कंप्लीटली सेल्स आर कवर्ड, कंप्लीटली सेल्स आर कवर्ड. यहां पर भी सेल है, यहां पर भी सेल है, चारों तरफ सेल आर पैक्ड. 3 डायमेंशनल स्ट्रक्चर. तो अभी क्या होगा समझो अच्छे से. तो क्या होगा? जो जस्ट बिलो द एपिडर्मिस, दिस इज द एपिडर्मिस, जस्ट बिलो द एपिडर्मिस, द सेल्स प्रेजेंट इन वर्टिकल रोज. वर्टिकल, दिस इज ए वर्टिकल रो, दिस इज ए वर्टिकल रो, and present just below the epidermis. This is another vertical row. You are looking at one cell, but at the lower side many cells are there. So this is a vertical row. This is another vertical row. This is another vertical row. तो चार vertical row अभी मैं दिखाया. तो अभी क्या होगा? जब टाइम बढ़ता जाएगा, the cells present in 4 vertical row become large in size, become different from others. उनका साइज बड़ा हो जाएगा. जो पहले जो सेल थे, सारे के सारे सेल सेम साइज का थे. लेकिन अभी ये चारों जो रो दिखाया, चारों रो जो कर्नर में दिखाया, these cells in 4 vertical rows become large in size with a large nucleus also. And due to that, the 4 corners are the 4 corners you find 4 vertical rows. तो अभी थोड़ा क्लियरली दिखा देता हूं और एक डायग्राम में. यहां पर और एक डायग्राम ले जाओ. दिस इज द आंथर. लोअर साइड क्या होगा? फिलामेंट. तो अभी ओनली 4 कर्नर का सेल दिखाऊंगा. 4 कर्नर थोड़ा डिफरेंट कलर में दिखा देंगे. यहां पर एक कर्नर था, यहां पर एक कर्नर था, यहां पर एक कर्नर था, यहां पर एक कर्नर. तो ये जो चारों कर्नर वर्टिकल रो है, ये एक एक सेल नहीं, रो ऑफ सेल्स. नीचे है रो ऑफ सेल्स, एंड दिस रो ऑफ सेल्स बिकम डिफरेंट फ्रॉम अदर सेल्स. अदर सेल को यहां पर नहीं दिखाया. अदर सेल जो छोटे छोटे होंगे, अभी वो सब बड़े होंगे. तो गेट डिफरेंशिएटेड. दिस vertical rows of cells just below the epidermis. कहा? Just below the epidermis. This is epidermis covering. तो just below the epidermis in 4 vertical rows cells are formed. and which vertical rows cells are called as archisporium or archisporial cells. Archisporium or archisporial cells. Archisporial cells. Okay, these cells are called as archisporial. तो आर्चिस्पोरियल कितना वर्टिकल रो का मिला? तो अगर क्वेश्चन आया आर्चिस्पोरियल सेल आंतर के अंदर आर्चिस्पोरियल सेल कितने मिलेंगे? 4 वर्टिकल रोज, नॉट 4. Don't say 4 archisporial cells are found. 4 rows. This is one row, this is another row, this is another row, this is another row. रो का मतलब बहुत सारे सेल इसके नीचे भी है, बहुत सारे सेल इसके ऊपर भी है, लेकिन ऊपर वाला पोर्शन हम हटा दिए. तो 4 rows of cells in vertical rows, 4 vertical rows just below the epidermis. These are called as archisporial cell, and they become different from other cells, get differentiated. So 4 vertical rows of cells at the corner just below the epidermis get differentiated, different from others. अभी हो गए आर्चिस्पोरियल सेल. लेकिन ये आर्चिस्पोरियल सेल क्या करेंगे? अभी थोड़ा पेरिक्लिनल डिवीजन शो करेंगे. पेरिक्लिनल डिवीजन का मतलब क्या है? थोड़ा इसको वर्टिकल रो में दिखा देता हूं. वर्टिकल रो, दिस इज अनदर वर्टिकल रो. ओके, 2 वर्टिकल रोज आर शोन. कभी ब्रेड का पैकेट देखो, ब्रेड पैकेट, एक ब्रेड पैकेट ले जाओ. ब्रेड पैकेट वर्टिकल रो में होता है. एक फर्स्ट ब्रेड, सेकंड ब्रेड, जैसे यहां पर दिखाया फर्स्ट ब्रेड, सेकंड ब्रेड, ऐसे. तो एक ब्रेड पैकेट भी एक वर्टिकल रो जैसा. एक ब्रेड पैकेट को ले जाओ और बीच में उसको वर्टिकली कट कर दो. वर्टिकली, नॉट ऐसे तो वो कट है. अगर ऐसे कट करेंगे उसको बोलते हैं पेरिक्लिनल डिवीजन. क्या बोलते हैं? पेरिक्लिनल डिवीजन. वो डिवीजन को बोलते हैं पेरिक्लिनल डिवीजन. और वो पेरिक्लिनल डिवीजन माइटोसिस ही होता है. पेरिक्लिनल डिवीजन क्या होता है? पेरिक्लिनल डिवीजन माइटोसिस ही होता है. और अगर वो पेरिक्लिनल डिवीजन हुआ, क्या होगा? तो पेरिक्लिनल डिवीजन हुआ तो अभी 4 वर्टिकल रो थे. अभी ये पेरिक्लिनल डिवीजन हुआ. अगर इसको ऐसे कट कर दिया, इसको भी ऐसे कट कर दिया मीन्स कंप्लीट. इसको भी कट कर दिया, इसको भी कट कर दिया, तो मेरा अभी क्या हो जाएगा? 8 वर्टिकल रो हो जाएंगे. 8 वर्टिकल रोज ऑफ सेल्स, 8 वर्टिकल रोज. तो नेक्स्ट डायग्राम में अगर दिखाएंगे तो नेक्स्ट डायग्राम में क्या होगा? देयर इज 8 वर्टिकल रोज ऑफ सेल्स. 8 vertical rows of cells. At the corner, each corner you find 8 vertical rows. One is towards outer side, another is towards inner side. Outer side, inner side. Outer side, inner side. Outer side, inner side. तो 2 रो बन जाएंगे. जो एक रो थे अभी 2 रो बन गए, तो 8 रो हो गया. लेकिन ये जो 2 रो बना, the outer cells, outer which are close to epidermis, ये वाला तो epidermis. Those cells which are close to epidermis, these are called as a primary parietal cell, and those which are away, those which are towards the center, this is the center, close to the center, these are called as a primary sporogenous cell. तो ये चीज तुमको याद रखना पड़ेगा. प्राइमरी पेरिटल सेल, प्राइमरी पेराइटल सेल, and the second cell is called as a primary sporogenous cell. Primary sporogenous cell, प्राइमरी स्पोरोजिनस सेल. ये चीज याद रखना पड़ेगा. तो ईच रो में, वर्टिकल रो में तुमको 2 सेल मिलेंगे, 2 वर्टिकल रो. प्राइमरी पेरिटल, प्राइमरी स्पोरोजिनस. यहां पर भी देखो, ये बाहर वाला हो जाएगा प्राइमरी पेरिटल, अंदर वाला हो जाएगा प्राइमरी स्पोरोजिनस. अगेन प्राइमरी पेरिटल, अंदर वाला हो जाएगा प्राइमरी स्पोरोजिनस. प्राइमरी पेरिटल, अंदर वाला हो जाएगा प्राइमरी स्पोरोजिनस. ये जो दिखाया प्राइमरी पेरिटल, प्राइमरी स्पोरोजिनस, ये धीरे धीरे चेंज होके क्या जो मैच्योर आंथर बन जाएंगे, जो मैच्योर आंथर दिखाया था. तो आज के क्लास के लिए इतना चीज तुम याद रखो. एक प्राइमरी पेरिटल सेल दिखे और एक प्राइमरी स्पोरोजिनस सेल देखे. तो उसके बाद हम देखेंगे वो कैसे प्राइमरी पेरिटल सेल और प्राइमरी स्पोरोजिनस सेल मिलके माइक्रोस्पोरांजियम बनाते हैं. तो नेक्स्ट क्लास हमारा रहेगा तो माइक्रोस्पोरांजियम कैसे फॉर्मेशन होता है. जो 4 माइक्रोस्पोरांजियम देखे थे, आंथर लोब एक साइड, ए वाला लोब हो जाता है, ए वाला और एक लोब हो जाता है. यहां पर एक माइक्रोस्पोरांजियम, ये जो टेट्रास्पोरांजियम स्ट्रक्चर कैसे होता है वो हम नेक्स्ट क्लास में डिटेल में पढ़ेंगे. तो दिस मच फॉर टुडेज क्लास. थैंक्स टू ऑल ऑफ यू.